Bismillahirrahmanirrahim. Today we are discussing development of limbs and limb musculature. Our objectives are explain the development of limbs, describe development of limb musculature with its innervation, and in the last we will discuss anomalies of the limbs or related to the development of limbs. So we start with development of limbs. So there is a term used as limb bud. Bud is a projection or out pocketing. So from the ventrolateral body wall at the end of fourth week of intrauterine life, the limb buds appear. So they are appearing at the end of fourth week as outpocketing from the ventrolateral body wall. And forelimb appears first, then the hind limb, means that our upper limb appears first, then the lower limb. The difference between both are 1 or 2 days. So the hind limb or the lower limb appearing on the bud of hind limb or lower limb is appearing 1 to 2 days later than the upper limb. What structures or which embryological germinal layers are forming limb buds? They are the mesoderm and ectoderm. A core of mesoderm or mesenchyme, which is derived from the parietal layer of lateral plate mesoderm, covered by the ectoderm. So here we can see there is a mesoderm, which is basically derived from the parietal layer or somatic layer of lateral plate mesoderm, is projecting from the ventral body wall, and it is overlapped by or covered by a layer of ectoderm, which is known as apical ectodermal ridge. Means that our limb buds are covered by ectoderm, and on the inner side of those buds we have mesoderm. So these mesenchymal tissue will going to form the bones and connective tissue of the limbs along with the musculature. So here we can see a embryo of 32 days, that is almost fifth week, and here you can appreciate that from the embryo ventrolateral body wall the buds or the projections are going out, which are covered by ectoderm, and inside we have mesoderm. Here we name it as upper limb bud, and in the lower part it is named as lower limb bud. These buds appear from the ventrolateral part of the body wall. Now, as we have discussed that the mesenchymal tissue, which is coming out as outpocketing from the body wall, it is overlapped by ectoderm, which is known as apical ectodermal ridge, because it is present on the apex. That's why it is known as apical. So, apical ectodermal ridge forms in the ectoderm at the distal border of the limb bud, as we can see. And the main function of this limb bud is that it forms a progressive zone or maintain a progressive zone, which is the region of highly proliferative mesenchyme and inhibits its differentiation. So it provides or maintain a progress zone. What is progress zone? The region of highly proliferative mesenchymal cells, and inhibits their differentiation. As apical ectodermal ridge moves distally, mesenchyme located more proximally undergoes differentiation. As it moves more distally, the mesenchymal tissue located more proximally undergo further differentiation. So this limb grows from proximal to distal, from proximal to distal direction. So this apical ectodermal ridge appear at the distal border. This is the distal border. This is the proximal part, which is attached to the ventral body wall. So here the progress zone is formed, which is a proliferative mesenchymal cells, And as this ectodermal ridge moves distally, these cells differentiated further, and the direction as we discussed is proximal to distal. The bone and cartilages differentiate from mesenchyme, and they are originating from lateral plate mesoderm as we have discussed. And the process of formation of bone and cartilage in the limbs are endochondral ossification. As in the foundation block we have discussed that we have three types of ossification: intramembranous ossification, intracartilaginous, or which is also known as endochondral ossification, and third is membranocartilaginous ossification. So here the limb bones are formed mainly by the intracartilaginous or endochondral ossification, which we can see here. Initially the mesenchymal tissue will form, and these mesenchymal cells will differentiate, condense and differentiate into the cells, bony cells, which are known as chondrocytes. Chondrocytes are the cells of the cartilage. So initially there is a mesenchymal cells, they begin and differentiate into chondrocytes, and these chondrocytes collect together, condense together and form a cartilaginous module in the middle. Then the blood vessels will invade into this cartilaginous module, and the chondrocytes will further changing into proliferating more and more, and osteoblasts are forming in the middle. And then as the blood vessel is moving more and more, blood supply is increasing, the osteoblasts are developing the bone from the center, and the cartilage will move toward the periphery or the end of the bone. So as we have discussed in the foundation block also, that a long bone has a central portion which is known as diaphysis, which is also known as the shaft of the bone, and the ends of the bones, the upper end and the lower end of the bone, are known as apophyses. And in between them there is a plate of cells which is known as metaphysis. So from here the chondrocytes toward the shaft side, that is diaphysis, undergo hypertrophy, and apoptosis will take place, and they mineralize and mix with the matrix. The osteoblasts, which are the bone forming cells, they bind to the mineralized matrix, and finally the blood invaded inside. So these all changes are taking place in the bone, and we name them as primary ossification centers. Primary ossification centers appear in the diaphysis or in the shaft, and this is happening that this primary ossification centers at the time of 12 week of intrauterine life, they all appear in the bones, and as they go further and further, ultimately the secondary ossification centers appears mostly after birth. Initially, mesenchyme will differentiate into cartilaginous module, the chondrocytes, they condense together, and then the osteoblasts are formed in the middle, and the cells which are present in the center, they die apoptotically and move toward the periphery, and the diaphysis completely mineralized with the bone. Then the ends contain secondary ossification center, which are going to be developed after birth, and these will form the epiphysis of the long bone, and the connection between the epiphysis and the diaphysis is the metaphysis. So this metaphysis is very much important because these is responsible for the length of the bone. So once the complete ossification takes place or the height reached, this chondrocytes will also change into the complete bone. So only we can see a epiphyseal line over here. Initially it is a growth plate or a pressure plate of cartilage, and then after complete ossification it becomes just a line. So that's how endochondral ossification takes place. So here also you can see a 6-week embryo, which is showing that hyaline cartilage is appearing So we can see that there are only pieces of cartilage initially which are going to develop into complete bone later on. So here you can see pieces of ilium, brevis, femur, tibia, fibula, also plate cartilages or the small bones of the foot which are going to form the foot bones and big toe as well as the fingers of the foot. Ultimately, with the tarsal and metatarsal bones. So initially, a cartilaginous model is formed, surrounded by the connective tissue and muscles, and then as they grow up forward, they form a complete bone. So what endochondral ossification we have discussed is taking place in between these long bones. And what happens initially when the mesenchymal tissue is present in the limb bud? It is completely present in the limb bud, but in the center these cells die apoptotically, and finally the gap will appear, and the gap will finally form the joints. So the mesenchymal tissue will differentiate into the ligaments of the joints, capsule of the joints, and the central part of the chondrocytes that are present in the initial model will die apoptotically, providing a joint space which is known as joint cavity. So that's how the joints are formed, and the bones are differentiated into different. That initially we have a single model, and then that single model, the cells within them start dying apoptotically, leaving the joint spaces, and the bones itself develop through endochondral ossification. So that's how the bones start to develop. That in the 6th week they appear like this, very small pieces, then a little bit enlarged in the further development. And beginning of the 8th week, you can see, you can appreciate the bones developing in their final shape. So in the 5th week or 4th week, the limb bud appears, and in the 5th week, the end of the limb is initially just like a paddle, flat area. As development proceeds, cell death occurs along the apical ectodermal ridge. In the 6th week of intrauterine life, the limb buds become flattened and they form the hand plates for the hand and foot plates for the foot. These limb buds, which have these flat structures known as hand and foot plates, separated from the proximal segment by a circular constriction. And a second constriction later also formed there, and dividing the proximal portion into two segments. Main parts of extremities can be recognized in the 6th week. You can see here, initially at the 5th week there is the limb bud projection and formation, but there is no constriction over here. So in the 6th week, the hand plate or paddle-like structure is formed, and first constriction appears over here. You can see initially it was straight, and then first constriction appears, which is separating the limb bud from this paddle ship area. This paddle ship area in future is going to form the hand in the upper limb and foot in the lower limb. So here you can see also, this is the foot, what we say, lower limb bud, and here is the part of the paddle ship foot, and this is the first constriction. And then up to the 8th week, the second contraction is very well defined at the end of the 6th week, and this diagram is of 8th week, but till 6th week at the end, there is another constriction appear over here which will form the complete part of the arm, and second constriction also over here. Initially it is a simple projection, then the first constriction occurs, and then a second constriction occurs. This will be the wrist joint in future, and this will be the elbow joint, and this proximal part is attached to the body wall, will be the shoulder joint. The same here, hip joint is attached to the body wall, knee joint and ankle joint. And this differentiation of these or appearance of these constrictions will occur 1 to 2 days later in the hand. So the fingers and the toes are formed when the cell death in the apical ectodermal ridge separates this ridge into 5 parts. The formation of the digits depends on their continued outgrowth under the influence occurrence of these five segments of village ectoderm, condensation of mesenchyme to form cartilaginous digit space, and depth of the intervening tissue between layers. How is this happening? That we will now. Then initially, these areas will start to die through the process of apoptosis, and this is the area of apical ectodermal ridges. So separate ridges are formed and cell death start to take place in between these clefts. So here you can see, here it was small but here is more areas of cell death. So the interdigit spaces are forming by the process of cell death. As the complete cell death takes place between the interdigital spaces or clefts, complete fingers are formed, and the digits operation is complete at the 56th phase of intrauterine life. That's how the fingers are formed. Initially, they are just like a hand plate and foot plate. So, just at their distal ends, the apical ectodermal ridges separates and cell death occurs in between them. And as it goes further and further, cell death of these ectodermal ridges, the lymph bones are formed finally with the fingers completely. In the seventh week of intrauterine life, the ridge is divided into five regions completely. As we can see, the between there is a cell death which separates the ridges as we have discussed. So, hand and foot bones are formed with the fingers. And another thing which is remarkable happening is changes in the position of the limb before the birth. So in the fifth week, when the limbers are appearing, they are appearing from the ventrolateral wall. So they have two ends. One is known as pre-axial and other is known as post-axial borders. This is the central axis of the limber, and this part will be pre-axial and this part will be post-axial. So what happens that as they are growing laterally, in the sixth week, limb bends anteriorly, so elbows and knee laterally, palms and so. So bending of the elbow as well as knee is taking place in the sixth week. In the seventh week, a very important thing is happening, that there is a rotation of limbs takes place along the long axis. We have discussed that this was the long axis, this was the long axis. So the limbs are rotated. The upper limb, which have pre-axial border over here and post-axial border over here, it rotates laterally. So the thumb appears lateral and little finger appears medial, and the flexor compartment is appearing on the anterior side. So our anterior compartment of the arm and forearm flexor because of the rotation at the time of development. So upper limb rotates laterally 90 degrees, and in the same way lower limb rotates medially. That's why the thumb of the big toe of the lower limb of the foot faces medially and the thumb is facing laterally because of this rotation. That upper limb along the long axis of the limb bud rotate 90 degrees laterally and lower limb rotate 90 degrees medially. And after final rotation, they come to their position of the adult life, and that is at the 8th week of intrauterine life. So in the 8th and 9th week, already endochondrial ossification begins and the blood vessels start to enter into the bony tissues. So here you can appreciate initially in the 5 weeks there is a liver, then in the 6th week there is a constriction appearing and the distal part becomes flattened like a paddle, and then in the 7th week apical ectodermal ridges start to form ridges, and these ridges die apoptotically, and finally the fingers are formed till the 8 week of intrauterine life with a proper rotation of the limbs. As we discussed, upper limb rotates 90 degrees laterally and lower limb rotates 90 degrees medially. That's why the flexor compartment of the upper limb is anterior and the flexor compartment of the lower limb is posterior. This is the diagram which is showing the hand and foot at the time of development till 5th week, and then in the 6th week, as these apical ectodermal ridges start to die through the process of apoptosis. Here you can see this area. So the bones and the tissue is going to form the fingers of the foot as well as the hand. So this pedal shape now is going to become a proper fingers. And here you can also see a 5th week embryo with limb appearing, then constriction appears, then the leaves appearing over here with a complete cell death. Finally, the 8th week of intrauterine life, the fingers are completely formed in the shape of the human being, and they are completely separated with the rotation taking place on the upper limb as well as the lower. That was about the development of the limb birds through upper and lower limbs, bone architecture and connective tissue. Now the few terms related to the muscle development or the limb musculature development. So myoblasts are the embryonic muscle cells which are derived from mesenchyme, and mesenchyme is what? Mesenchyme is the embryonic connective tissue. It forms the embryonic connective tissue and we name it as mesenchyme. So whenever we are talking about mesenchyme, it means we are talking about embryonic connective tissue. Other terms which are related to muscle development are already you have talked about in the embryology and foundation, somite. At the end of the 3rd week of intrauterine life, paraxial mesoderm forms a segmented series of tissue blocks on each side of the neural tube. They are known as somatomeres in the head region, and somites from the occipital to the sacral region. Actually, what is a somite? It is a segmented portion of paraxial mesoderm, and we name somatomeres in the head region and somites in the rest region from occipital to sacral region. Another term we use myotome. Myo means muscle and tome means a portion or segment. So a muscle or a group of muscle derived from a one somite and innervated by a single segment of a spinal nerve. That is known as a myotome. So myotome, a muscle or a group of a muscle from the derived from one somite and innervated by a single segment of the spinal nerve. So important thing is there that every muscle which is forming through a myotome, they are carrying their own segmental supply of the spinal nerve of the corresponding region. So here you can see the formation of somites appearing first pairs and then here appearing around the neural tube as a whole. So each somite is actually a part of paraxial mesoderm, and from here, from these somites, the musculature of the limb will appear. So also you can see in the lateral view, these are the musculature of the myotomes which are appearing in the occipital region, cervical region, thoracic region, and so on in the lower region also. And this is our limb birds. So that part of the myotomes which are present in the region of the limb birds or just opposite the limb birds will going to give formation to the muscles. So muscles of the upper limb will appear from this region segments of muscles, and the same areas or segments of nerves are supplying these muscles. So the limb musculature in the 7th week of intrauterine life, limb musculature is developed from mesenchyme near the base of the limb bud as we have seen in the previous picture. The mesenchyme is derived from the dorsolateral cells of the somites. So from the Somites are divisible into parts, so their dorsolateral cells are mainly responsible for the formation of the limb musculature cells. With the elongation of the limb bursts, the muscle tissue splits into flexor and extensor components and enter into the limb bursts. I will show you the other picture. So the upper limb bursts lie opposite the lower 5 cervical and upper 2 thoracic segments, and lower limb bursts lie opposite the lower 4 lumbar and upper 2 sacral segments. And from these segmental myotomes, the muscles as well as the nerves are coming to supply the upper limb as well as lower limb. So here you can see, as we have seen before, that we have limb bud appearing, and there are myotomes which are C5, C6, C7, C8, and T1, T2 mainly. These are which are going to give the musculature as well as the nerve. And as the musculature is divided, forming when they enter into the limb bud. This is a limb bud from the body bone. This is the musculature into the body bone. This is a limb bud. As they are starting to penetrate into the limb bud across the bone, they split into extensor muscles as well as they split into flexor muscles. So extensor as well as flexor muscles are split at the time of development when they are starting and forming and entering or penetrating into the limb bud. And also when they penetrate, they carry their nerve. So as we already know, the spinal nerve is formed by the medial and ventral and dorsal root, and then they divide into ventral division and dorsal division. So the ventral divisions, when they move forward toward the ventral body wall, they give branches to the muscles. So important thing is that every muscle, when it is moving and forming into the limb, upper and lower limb bursts, or they are coming inside, they carry their own nerves together. That's why every muscle has they have their expressiveness in them. So which nerve is supplying the muscle is the depending upon the formation of the muscle at the time of development. So regarding the limb innervation, we have sensory innervation as well as we have motor. Motor is the muscle supply and sensory for the skin. So C5, C6, C7, C8, T1 and 2, as already we have discussed, that these are the segments which opposite to them are the limb buds appearing in the upper limb. So these nerves are entering into the upper limb and they are responsible to supply the upper limb, their musculature as well as their sensory supply. Here you can see their arrangement and the anterior view. You can see C5, C6, C7, C8, T1 and T2. So as the limbs are appearing, the nerves are migrating from the spinal segments toward the upper limb, and the same is happening in the lower limb. So here you can also appreciate the upper limb segments and lower limb segments. Only these nerves are supplying upper limb and lower limb from the all spinal nerves. As we have discussed that the border which is upward over here, this one or this one is a preaxial and this one is a postaxial border. As they are developing further, the nerves are moving toward the preaxial and postaxial depending upon whether they are sent to supply the flexors or whether they have to supply the extensors. So here in the upper limb you can see preaxial border and postaxial border. Facial is another entry. And as the rotation takes place, already we have discussed that in the 7th week of intrauterine life, rotation of the limbs takes place. So flexor compartment appears in the upper limb anteriorly and extensor compartment in the lower limb appears anteriorly because of their rotation. So that's how the nerves through the segments they enter into the upper and lower limb for the motor supply as well as for the sensory supply. And for the sensory supply, we use a term which is known as dermatome. Derma means skin and tome means part. So part of a skin, dermatome means a part of a skin supplied by a single spinal segment. So sensory innervation to the limb maintains a segmental pattern reflecting the embryological origin of the dermatome. So how these nerves are coming the same way as they start to penetrate from the spinal segments to the body wall and the limb, they decide their region of supply. So these are the segmental supply of the skin, and name this as sensory supply or the dermatomes. Dermatome, part of the skin supplied by a single spinal nerve. This is the anterior view. Nerve which are supplying anteriorly. C4 at the shoulder, C5 in the middle of the forearm and arm, and lateral side is C6. Lateral two fingers C7. Medial fingers with the medial part of the arm forearm is C8, and then a small strip between these are for the frontal area is T1, and finally at the alveoli is T2. The same effect on the posterior side. That's how the supply. Now we will talk about some anomalies of the limbs. The anomalies of the limbs, if there is a complete absence of the limb due to male development, that condition is known as amelia. And if there is a presence of limbs but they are not completely formed or partial presence of limb is there, this will be known as meromelia. So meromelia is a partial formation of the limbs and amelia is absence of the limb. Why it happened? Mainly it happened due to genetic factors and also a very important medicine which is known as thalidomide, which usually given during pregnancy for the use as a anti-nauseating or to prevent vomiting, also used as an anti-cancer drug, and already we discussed anti-nausea drug given for the morning sickness between the 1950s and 60s. Exactly during the 1957 and 1962 period, that is 5 years of that period, most of the babies developed amelia or meromelia because of the side effects of this medicine, which is known as thalidomide. So in 57 to 62 years, at that time, many patients appearing as amelia and meromelia. So after the research, they have identified that this medicine, which is thalidomide, causes damage to the development at the time of organogenesis period and causing these problems. Another type of meromelia is phocomelia, in which you can see that there is no arm and forearm, only the foot plates or hand plates, which are also not complete, they are directly attached to the trunk, as you can see. So the limbs are not there, but the hand and foot are attached directly to the body. Their condition is known as phocomelia. Another term we use micromelia, in which the limbs are present but they are smaller in size as compared to the normal structure. That is known as micromelia. So amelia, complete absence of the limb. Meromelia, partial absence of the limb. Phocomelia, limbs will not be there but hand and foot will directly attach to the trunk. And micromelia, limbs are there but they are smaller than the usual size. Other anomalies of the limbs are related to the digits. Some people have extra digits. Again, this is happening that when the apical ectodermal ridge is forming, which is between or cleft between the fingers. If they form one extra cleft, then an extra digit will form, and this condition is known as polydactyly. So polydactyly means extra digits. Another condition is known as synpolydactyly, extra digits and fused digits. What are that? I will tell you. After polydactyly, we have a term used syndactyly. Syndactyly means digits are 5 but they are fused together. Because of the fused formation, the number of the digits will decrease, and this is known as syndactyly. And sometimes patient has both syn polydactyly, they have extra digits but their digits are fused together. So we have polydactyly, extra digits, syndactyly, fused digits, synpolydactyly, fused and extra digits, and then we have a term which is used as cleft foot or hand, or also known as lobster claw commonly, in which always there is an absence of third metatarsal or metacarpal bone, because there is no metatarsal or metacarpal bone. So first and second digits and fourth and fifth digits fuse together. First and second, third and fourth fuse together, and in between there is a cleft. big cleft and they appearing as a lobster claw known as cleft foot or cleft hand. Here you can also see this is the third metatarsal or metacarpal bone is completely absent. So the first two and the fourth and fifth digits unite together or fuse together. So only one cleft is formed in between them, and that condition is known as cleft foot or cleft hand. This diagram is of cleft foot. Already we talked about fused digits, which is known as syndactyly. So this diagram is showing that index finger is okay, but the rest of the digits are fused. Over here index and little fingers are okay, but middle two fingers are fused. And here you can see another term used, radically. Radically means that the fingers are there but their length is shortened. If the digits are short, then we name them as radically, and if we have extra digit, we will name it as polydactyly. Another condition which is known as osteogenic imperfecta, a baby with bone with the short bones, hypomineralization of the bones, as well as bowing of the limbs. The limbs are bending, shortening, and this condition is known as osteogenesis imperfecta. You will talk more about this in clinical science. Then in some babies what happens that few of the fingers are normal but some of the fingers are not completely formed, as you can see, or abnormal fingers are there, which is known as amputation. This digit amputation occurs due to the amniotic band. So what happens that at the time of development, these amniotic bands are formed and these amniotic bands tie around these fingers. As they tie around these fingers, they constrict these fingers and they block their blood supply. As the blood supply is blocked, the amputation will take place and they will not further form and they will damage it. So constriction will be there because of the presence of amniotic bands. So either there will be constrictions, not completely proper forming of the fingers because the bands are so encircled around it and constricted, or sometimes they completely damage it. So blood will not pass forward and the area will not be there, it will be amputated. This is known as digit amputation due to the amniotic band form. Another term condition where is a transverse limb deficiency. It may occur if there is a disruption or dysfunctioning at the level of apical ectodermal ridge, which is not completely or properly forming, or maybe due to the vascular abnormalities, the blood is not properly reaching there. So there will be a transverse limb deficiency when it appears like this. And another condition is known as congenital hip dislocation, in which the acetabulum and the femur head is not fixed properly and they are dislocated since the time of birth. And how it is identified that when they move the hip joint, there is a clicking sound appearing. So it is surgically treated. That is known as congenital hip dislocation. This is all about the anomalies of the limb. So if you have any question, please ask. Thank you very much.